So back to DNA and DNA structure. As I mentioned, Watson and Crick were two theoretical biologists who modeled the DNA structure based on the X-ray images of Rosalind Franklin and the base composition work of Edwin Chargaff. They were able to deduce that, first of all, that the bases inside DNA were forming hydrogen bonds between each other in order to stabilize the structure, and that the bases were facing inwards as opposed to outwards, like the first structure proposed. That adenine and thymine were forming only two bases, two hydrogen bonds between each other, while guanine and cytosine formed three. That the DNA was a double helix due to the degrees of freedom that the ribose and the phosphates allowed for the DNA, and that it was oriented in the anti-parallel fashion. We'll discuss that a little bit later. Here, I wanted to show you the actual three-dimensional structure of DNA on JMO. Just a second. So as you guys see here, in bright purple, is the short stretch of a double-stranded DNA helix. As I rotate, this is the more familiar picture that the textbooks display, and as I rotate the image around itself, the model around itself, you can appreciate that there are two grooves inside the DNA, one that's slightly wider and the other that's slightly narrower. So between here and here is the minor groove of the DNA, and this and here is the major groove. And if you guys remember, the major groove of the DNA is where the transcription factors tend to bind, because these base pairs are a lot more accessible and you can form hydrogen bonds to their sides. The other important thing is the base pair stacking that you can see occurring in this DNA molecule. So I hope you can appreciate how perfectly parallel and flat the bases are when they stack inside the molecule. So you go, there's your thymine and your adenosine, thymidine and your adenosine. There's guanine and cytosine forming base pair there. And you can draw lines through them and they will be perfectly parallel. And this base stacking, because the bases themselves are hydrophobic to a certain extent, this very precise parallel base stacking contributes to the rigidity, to the structure of the DNA. It makes it a lot more rigid, a lot more stable. Sorry, that probably is the best word. So base pair stacking contributes to the stability of the DNA molecule. And then the final view I wanted to show you guys is how perfect of a helix it really is. You can see that if you look right through the molecule, you can see how the width of the helix barely changes, and that all base pairs are aligned inside of it in a very particular fashion. So I'm sure you have realized here that while the base pairs themselves are shown in the molecule, the pictures for the riboses and the phosphates have been completely removed. So the actual helix is slightly different, but I think this is a really pretty way to look at it. So things to retain is, from the structure, is that the base pair stacking, the very parallel base pair stacking, contributes to stability, and then there's two grooves inside of the DNA helix, the wider major groove and the narrow minor groove. Okay, back to lecture. Sorry. What I also wanted to say is that, first of all, by clicking on the link, you can go to this picture. Also, that the Protein Data Bank is a freely available website that contains protein structures and RNA structure for all kinds of proteins. And as long as you know the code, which is a 4-letter code for a particular protein, or you just use their search function, you can look out how the RNA polymerase works, how the helicase works, how a transcription factor of your choice binds the DNA, deforms DNA, etc. It's a very broad resource, and I encourage you, as you guys learn about more and more different proteins that participate in different molecular biology mechanisms, that you take the advantage of the modern age and look up the three-dimensional structures of these proteins available in PDB. Because I think it will help you learn more about the mechanism visually as opposed to just listening to me talk. Because there's only so much I can cover. So I encourage you to be naturally curious, because that is molecular biology is a path that you chose, and it's a scientifically rigorous discipline. And if you're not curious about it, you will not advance very far. All right. After this pep talk, let's talk about directionality. So both DNA and RNA molecules have directionality, simply because if you just look at one base here, one nucleotide here, which has the phosphate, has the deoxyribose, and has the base, it hasn't been drawn on here, it has directionality. The phosphate is attached to the 5' carbon, and the ribose, the other carbon that will participate in the formation of the phosphodiester bonds that will form the DNA or the RNA molecule, is the 3' carbon. Because of that, and because of the fact that DNA polymerases and RNA polymerases synthesize DNA only in one, or RNA only in one direction, they always go 5' phosphate to 3' OH. That is a standard way to look at a DNA or RNA molecule. It's not just boring nomenclature. That's really important, because all of the technologies that you guys hopefully want to learn about as a molecular biologist, such as primer design, sequencing, cloning, protein expression, you name it, will rely on your understanding that a DNA molecule or an RNA molecule has directionality, and that it starts at 5' and it ends at 3', even though we count 1, 2, 3, 4, 5 the other direction. And why it's 5' to 3'? Because the phosphate is attached to the 5' carbon. And then the next phosphate in a nucleotide chain will be attached to the 3' carbon of the deoxyribose. So hopefully I said 5' to 3' enough times that we will remember it forever, because it will come up. Because DNA and RNA molecules are directional, there's several ways you potentially could be winding that double helix. And the way the DNA double helix is wound, as well as the RNA double helix, when it happens to have a double helix, is in the anti-parallel fashion, meaning the top strand is going to go 5', 3' this way, and the bottom strand is going to go 5', 3' that way. As in pictures here. So you can see the top strand has a 5' phosphate on this side and the 3' OH here, and the bottom strand of this flattened out DNA double helix is oriented in the opposite direction. Or if you look at the actual molecule, the image that we saw earlier, you can see that this side of the helix is going 5' to 3' this way, and 5' to 3', and the other guy is going anti-parallel to it. Also, because the double helical structure of the DNA or a double helical RNA are only held together by the hydrogen bonds of the base pairs, there's no covalent attachments there, remember? The hydrogen bonds can be broken when the molecule of DNA or RNA is either heated, which gives every single molecule inside the DNA more energy, and entropy and heat will break apart the DNA double helix. Or if you provide the hydrogen bonding atoms something other than each other to hydrogen bond with such an alkaline solution where there's other hydrogen bonds that can be formed with the actual with the actual base, that will also cause denaturation of the or acidic, can be alkaline, can be acidic. That will also cause the breaking of the double helical structure of the DNA and allow for the single DNA molecules to float around in solution. An important thing here is that to know is that these arrows are going both ways. You can see that denaturation and annealing both can occur. So after you have removed the DNA out of its alkaline environment, acidic environment, or the high temperature environment, the double helix will refold itself. It will take, for a very long molecule, it will take many tries to get it perfect, but in the end, it will become this perfect double helical DNA again. And the graph that just appeared on the screen just shows the graphical, instead of a graphical, it's as opposed to the pictorial representation of what I'm talking about. Because when you read scientific papers, nobody's gonna, draw pictures for you. It's not a cartoon. So you can see that the double-stranded DNA is converted to a single-stranded DNA as the temperature increases here, and a particular point, just like KD, just like KM, there's also melting temperature, standard melting temperature for different types of DNA. That's why 50% of the double-stranded DNA have been denatured and are now separate. Oops. TM can be calculated, and so the length and the GC composition of the different DNA molecules will affect when those particular double-stranded molecules will separate, aka melt. Also, I want to call you guys' attention to the fact that on the y-axis there is this mysterious notation called A260. What it says is that A stands for absorption, and 260 stands for the wavelengths of light. This is 260 nanometer light. Because DNA has so many bases inside of it, it reacts with, it's able to absorb ultraviolet light of the 260 wavelengths and the 280 wavelengths. And that, because absorbance of light is easy to measure in a machine called a spectrophotometer, reading the absorbance of a DNA or an RNA solution is one of the standard ways that we measure how much DNA or RNA is in that solution. All right, so let's recap. We know now that DNA and RNA have orientation. They are oriented 5' to 3'. Because they're oriented 5' to 3', because they're also made, they're synthesized 5' to 3', they have they're arranged antiparallel when they are in a double helical situation. That the double helix can be broken down into single strands easily because the double helix is only held together by hydrogen bonds. So if you heat or change the pH of solution, you will be able to pull apart the two strands. Another important characteristic of the DNA and RNA comes from its backbone. Because the DNA and RNA is held together by phosphodiester bonds carry a partial negative charge because they have this oxygen here that stole stuff from a phosphate and is partially charged. So in cells, the negative charge of the DNA or RNA molecule is neutralized by metal ions that will bind to the phosphate, or by positively charged proteins such as histones that coat the DNA and spool it into nice little spools. In water, when the magnesium ions or the histones have been stripped, water will start forming hydrogen bonds with the phosphodiester bonds, creating this cloud of water molecules around DNA, and thus making DNA very, very soluble in water. However, one can use the fact that DNA and RNA is negatively charged, and by replacing the water molecules that are in this picture are these little dots that are coating the DNA structure. If you replace the majority of the water surrounding a DNA molecule with salts, with salt ions that will also interact with a phosphate backbone, and then suddenly change water to ethanol, you will be able to precipitate the DNA out, which is exactly what happens in this picture. So here, a small amount of salt has been added to water, and majority of the molecules around the DNA here have been replaced by salt. And when ethanol, you can see how much more ethanol, this whole tube is filled up with ethanol, was added. The rest of the water is displaced, and the ethanol is not the strong dipole that water is. As a result, the DNA, because it's a very large molecule, it actually should not at all be soluble in water if it didn't have these negative charges on its phosphate. The DNA crashes out, and you can see it. It becomes this milky, goopy cloud. And this process of changing the solubility of DNA between water and ethanol by adding a little bit of salt to remove the DNA